This animation will show the various sequencing methods used in next-generation sequencing. Method 1. Pyrosequencing. The library fragment is placed in a vessel with DNA polymerase and washed with a single nucleotide. The complementary nucleotide is incorporated into the synthesizing strand. This releases a pyrophosphate. The pyrophosphate and adenosine phosphosulfate are catalyzed by sulfurylase to produce ATP and sulfate. The ATP in turn reacts with luciferin and is catalyzed by luciferase, producing oxyluciferin. Upon this reaction, light is emitted, which helps identify the exact base in the sequence. The intensity of light recorded corresponds to the number of nucleotides incorporated. The unreacted nucleotides are removed by the apyrase enzyme, and the next nucleotide is added to identify the next base. The process is repeated until the whole fragment is sequenced. Method 2. Sequencing by synthesis. A mixture of fluorescently labeled nucleotides is washed over the vessel containing the library fragments. Only one nucleotide is incorporated in each cycle due to the terminator present on the 3' end. Light is emitted when the nucleotide is incorporated, and the wavelength of emitted light reveals which nucleotide was incorporated. Next, the fluorophore and the 3' terminator end are cleaved and removed. The cycle is repeated till the whole template is sequenced. Method 3. Ion semiconductor sequencing. The process occurs within a microwell with an ion-sensitive semiconductor plate below it. Single nucleotides are washed over the vessel, and the complementary nucleotide is incorporated into the template strand. This releases H+ ions, changing the pH of the solution. The proton semiconductor measures the pH change and calls the base which is incorporated. If there is no incorporation, there is no pH change. Method 4. Sequencing by ligation. This method relies on the incorporation of oligonucleotide probes by DNA ligase. The probe has 2 specific bases, 6 degenerate bases, and 1 of the 4 fluorescent dyes at the 5' end. In the first round, a primer of length N is used. The fragment is exposed to a library of 16 probes. An appropriate probe is added to the primer using DNA ligase. Fluorescent light emitted is recorded, and the last 3 degenerate bases are cleaved along with the fluorescent label to allow the next probe to be ligated. The cycle of ligation is continued 7 times to form a chain. The second round uses a shorter primer, and the process is repeated. A shorter primer is used for each round until every base is identified. The last method is nanopore sequencing, which is classified under 3rd generation sequencing and does not require base incorporation. In this method, DNA is passed through a nanopore across a membrane. The DNA helix is first unzipped by an enzyme. The single strand can then be driven through the nanopore. Each base reduces the flow of ions to a different degree. Thus, based on the change in current, corresponding DNA base is identified.